did not show clinical signs, but had viable MAP in intestinal tissues as well, and in diaphragm muscle as well, and that both of them were beef cattle. So these results suggest that symptomatic animals, but also as in asymptomatic animals, the bacteria can disseminate from the gastrointestinal tract to the muscle tissue. And finally, we also wanted to look at those animals and see if we can detect the MAP in feces of them. And we tested 4 of the animals and 3 were positives. So this also agrees with the idea that the detection of MAP, the chances or the likelihood of finding MAP is higher at the late stage of infection when the animals are actually setting the bacteria to the environment. And, and then finally we also check the blood of these animals, and we just found MAP in the blood of one of the animals. MAP was found in blood of only one of the 6 animals, showing evidence of MAP in diaphragm muscle. And so this might occur because the delivery of the infected macrophages to the blood is a transient process, and it doesn't occur continuously, so it is really hard to detect the bacteria in the blood of the animals. So, as conclusion, I'd like to conclude that MAP, this is the first report showing MAP and detecting and culturing MAP from diaphragm muscle of paratuberculosis infected cattle, this time for human consumption. Second, we were able to see an association between the dissemination of MAP and severe enteric lesions, clinical signs, heavy bacterial load, and fecal setting of MAP. And third, 2 of the positive animals did not show clinical signs associated to paratuberculosis, suggesting that meat from both symptomatic and asymptomatic animals could potentially represent a source of exposure of MAP to humans. And, whoops. Okay. That's going too fast.
Go back one more?
Yeah, please. Yeah. Okay. All right. So, next. All right. Although this is a very preliminary study, and we have surveyed 47 animals, we are planning to test more skeletal muscles commonly included in ground meat products for the presence or absence of MAP. And we also think it's very important to clarify what is the risk of exposure of humans to MAP via contaminated meat. While heating meat might be very effective in reducing the amount of the bacteria, no previous studies have shown the temperature and the time that is required to reduce the load of the bacteria when present in a meat matrix. And third, we think that it would be very important to develop effective ante-mortem methods for the identification of cattle with disseminated infection, especially for the animals that are subclinically infected with the disease. And previous studies have shown that a large percent of animals that had disseminated infection in other organs escaped the detection by serum ELISA or liver biopsy. So new methods and sensitive diagnosis methods should be developed to detect subclinically infected animals. And so while all these studies are underway, and the food and cattle industry should adopt a precautionary approach and implement more control measures to reduce the chance of MAP contamination of foods and the entry of MAP-infected animals in the human food chains. And this is really important, especially in the case of the test and cool programs, because the animals that are positive after those programs, they are sent to a slaughter, and the meat, if the meat reaches the standards, it can enter easily the human food chain. And so finally, this study has been very recently published in Foodborne Pathogens and Disease, and I would like to thank Dr. Ramon Juste and his paratuberculosis research group and Michael Ternalia for their continued support, especially to Elena Molina, that helped with the analysis of the data, the cattle owners, and all the veterinarians and staff of the slaughterhouses that participated in this study, the histopathology department, and finally to say that the study was funded by grants from the National Institute of Agricultural Research and the Department of Science and Innovation from Spain. And thank you all for your attention.
All right, we have 5 minutes for questions, but if anybody runs early, there is a talk at 10:30. The schedule was wrong. So you have a half-hour coffee break, and then Ramon is going to be speaking. But we still have room for questions right now. Yes?
Can you explain once again how you distinguish between disseminated infection, post-mortem spread due to the breakdown of the entire body of the blood supply of the intestinal tissues, and contamination during the slaughtering process?
Okay.
So the question was, how can you distinguish, I guess, an antemortem spread, a breakdown at necropsy, and post-mortem spread, presumably during the sample procurement? Is that the question?
Yeah, that's a very good question. And we were very careful with that because what we did was we take first the sample from the diaphragm muscle to avoid cross-contamination with gastrointestinal tissues. So we took first the sample from the diaphragm, and then in the P3 lab we took samples from the gastrointestinal tissues, and we were very careful to try to avoid any contamination or carrying any kind of contamination when we collected the samples.
Okay, but is that sorry?
A follow-up?
Yeah, the animals were regularly slaughtered. The animals were hanging out with their rear legs, and the intestines have to be removed. And then you have fragments of muscle below the intestines. Can you simply rule out that the material has fallen down in the muscle and contaminated the muscle from the outside?
So according to the questioner, the animal is hanging upside down from its hind legs. It's been slaughtered, the intestines are out. Before the diaphragm was procured, or was the diaphragm procured before the intestine was touched?
Yeah, we took an internal sample of the diaphragm muscle, okay?
Question here?
Sorry, from what?
Have you collected samples from vital organs such as the liver, kidney, perhaps spleen, other organs?
No, not in this study. No. That has been previously proved in other organs. Dissemination of mass from the gastrointestinal tract to liver, spleen, genital organs have been already proved, and we just look at first if we can detect in muscle tissue.
I believe there's a question from Timble.
Do you know of the location of the tissue of the organ?
Do you know the location within the tissue? Presumably you're asking the cellular location. Was it extracellular, intracellular, which type of cell type, or any information about the infection within the tissue?
Or is it associated with nervous tissue?
Or nervous tissue?
No, we haven't done that, no.
You had one that you said was in the blood. Was that culture positive in the blood with colonies on a plate, or was that positive by PCR or the real-time PCR?
No, it was by culture from the blood.
With colonies on the agar?
Yeah.
Okay. Yes?
Were any of these animals lactating? Were you able to get milk? Were you culturing the organism out of milk?
No, we didn't test milk.
The question is whether
that's
any of the
good
animals were
question.
lactating.
That's good question. No, we didn't do that.
Excellent, Marta. I'd like to thank the 3 speakers for being on time.